Welcome to Biology My Passion. I am Soumya Harikrishna. We are dealing with the chapter Cell Cycle and Cell Division. Now, if all the preparations are done in interphase, then comes the mitotic phase or M phase, where the actual division happens. So, in M phase, there are two stages, karyokinesis and cytokinesis. Karyo means nucleus. So, first the division of nucleus takes place, then followed by that cytokinesis, division of cytoplasm. Is there any problem if we reverse the sequence, first cytokinesis and then karyokinesis? Think about it. Yes, if first the cell has one nucleus, first cytokinesis happens or the cytoplasm divides, we know one daughter cell will have nucleus, the other one will not have. This will not exist, right? It will die off without nucleus. Only the one with nucleus will exist. Do we need only one daughter cell or two? We need two. So first what should happen? Inside the cell, the nucleus should divide into two. Then the cytoplasm divides. As a result, each daughter cell will have one nucleus each. Now we will learn the details of this karyokinesis of a M phase. Let's now dive into the details of M phase, in which again the first phase, karyokinesis. Karyokinesis involves basically four stages. First is prophase, second metaphase, third anaphase, and fourth telophase. So first a prophase starts with a P. So if you take only the first letters, we can remember as PMAT. Usually we remember like that, PMAT. You can make mnemonics out of it. If you have very good mnemonics, interesting and easy to remember, and you like to share with other friends of your age group, you can post it in the comment box, which will help many people. Okay, so prophase, metaphase, anaphase, and telophase, PMAT. Always remember the sequence, is very important. You cannot say metaphase, then telophase, or prophase, anaphase, that's wrong. Okay, it is in a sequential manner it is happening. So in prophase what happens? So initially now suppose this is the cell and here is the nucleus. Inside what is there? The DNA is there, right? The DNA strands are there. But these DNA strands have already duplicated. So a duplicated one I am drawing with a red. That is an extra copy. But the number is the same. So how many DNA strands I have drawn here? Only 4 here, right? 2 pairs we can say. Now at the time of prophase, these thread-like structures will be now condensing to form the typical chromosome. So these threads will condense and form the chromosome. They condense like this. I am drawing only one arm because the other one I will draw with the red. Okay. So there is a central part, that is the centromere, which is the primary construction on a chromosome. So this is a condensed form of chromosome with the 2 chromatids. So during prophase, what is the first stage? Chromatin materials condense to form rod-shaped chromosomes. And one more thing happens here. We learned the centrioles duplicate during the S phase of the interphase. Those centrioles now will move to the 2 opposite poles of the cell. So here they will reach on both the poles, and from there small microtubules will come out. These microtubules are forming aster. And connecting these two daughter cells, there are daughter centrioles, there are spindle-like fibers, they are called spindle fibers. Now, what happens? The centrioles move to opposite poles. That's the second thing happening here. And also the spindle formation begins. And third one is, by the end of this prophase, the nuclear envelope or the nuclear membrane along with the other organelles like endoplasmic reticulum, Golgi apparatus all disappear. They will not be found in the cell during the end of prophase stage. So prophase mainly 3 events are happening. First, a chromatin material which are thread-like condensed to form rod-shaped chromosomes. At the same time, the duplicated centrioles move to the opposite poles and start forming aster and also the spindle fibers. Those spindle fibers and aster together we call it as spindle apparatus. And finally, what is happening at the end of prophase, nuclear envelope, nucleolus, Golgi apparatus and endoplasmic reticulum, nucleolus also, they disappear. This is what is happening during the first stage or the prophase. The longest phase in mitosis is prophase. Now let's move on to the metaphase. So when there is a disappearance of the nuclear membrane at the end of the prophase, there starts the metaphase. During metaphase, the chromosome condenses to its maximum. So if anybody wants to learn which is the bestest stage of studying the chromosome structure, they have to choose metaphase stage. Now during metaphase, what happens? All the chromosomes have centromere region where kinetochores are there. So the spindle fibers will come and attach to the kinetochore. And so these are the spindle fibers. Suppose this is one chromosome, here is another chromosome, here is another chromosome and here is another chromosome. So these spindle fibers, they come and attach to the kinetochore on both the sides. Okay. And now after this there happens a tug of war. As a result, everything will be brought to the center. So all chromosomes in the cell will come and align at the equator region. So that is the important stage or how we identify metaphase. So how is metaphase identified? All the chromosomes will come and align at the equator. The centromere will be in the center, right? Rest of the arms can go in different directions, but all centromeres in the center. So two things happen. First, spindle fibers attached to the kinetochore and then chromosomes are aligned at the equator. Now, one more thing to remember here. In the case of higher plants, astral formation is not happening. So it's called anastral mitosis. Whereas in animals, astral formation happens. So it is called astral mitosis. Now we move on to the next stage, that is called anaphase. Anaphase is the longest phase. During this time, what happens is these spindle fibers, they start shrinking or condensing. As a result, they pull this kinetochore also towards them because it is attached. As a result, what happens? These chromosomes, centromere region will split apart and the chromatids separate. So one half of the chromosome will go to this. That means that was the actual content. We know the second was the duplicated one. So one copy goes here, another copy is here. So these are called daughter chromosomes, but we say the chromatids of a chromosome separate. This is very important term you have to use. Don't write chromosome separate. Okay. During anaphase, chromatids of a chromosome separate. So suppose 46 chromosomes are there. All 46 will come at the center. Every single one is split into two. So one set of 46 will go to one side. One set of 46 will go to the other side. So ultimately how many we have? 46 on each side. Here also we started with 4. Now also we have 4 on both sides. Now coming to telophase, what happens? This will come to the pole and they again decondense. The rod-shaped condensation is lost. Again it will form thread-like structures like the chromatin material, and that marks the telophase. Here actually what is happening are the opposite of what happened during prophase. That is chromatin condensed to form chromosome here. Here chromosome decondensed to form chromatids. Then centrioles move to the opposite poles. Here the centrioles disappear. And nuclear membrane, endoplasmic reticulum, Golgi apparatus, nucleolus, everything reappear. Okay, so this is telophase. After telophase, you will see 2 nuclei in one cell. Please do share my videos with all the friends so that you can help me reach more audience. That will make a lot of difference in my content creation. Please like, share and subscribe to my channel. Biology, my passion. Thank you for watching.